Hello students, welcome to SK Biology. In our previous lectures, we have discussed discovery of genetic material and quest by different scientists to understand genetic material. With the efforts of all scientists, we came to know that DNA is a genetic material. But now important question is how DNA maintains transfer of genetic information from one generation to next generation. How DNA controls heredity. This question was answered by scientists Meselson and Stahl. By density gradient centrifugation, they discovered that DNA replication is semi-conservative. Let us understand their experimental details. In 1958, Meselson and Stahl studied replication process of DNA, in which they found that semi-conservative is the process by which DNA duplicates itself before cell division, ensuring that genetic information is accurately passed to daughter cells. In semi-conservative replication, double-stranded DNA helix is separated into two single-stranded molecules, and these single-stranded molecules acts as a template on which new DNA strand is synthesized, which results into production of two identical DNA molecules, each containing one new and one old DNA strand. Now we will learn how Meselson and Stahl proved this replication model. Meselson and Stahl studied composition of DNA, from where they came to know that DNA is made up of nitrogenous bases. That is, nitrogen atom is a building block of DNA. They know that nitrogen has different isotopes. Meselson and Stahl used two isotopes of nitrogen for their study. One isotope is called as light isotope with N14, in which 7 electrons, 7 protons and 7 neutrons are present. Here in this image, protons has been shown as red, electrons are green and neutrons are blue in color. Another isotope they have used is heavy isotope of nitrogen N15, in which one extra neutron is present which increases its weight. That is, N14 is lighter than that of N15. This light and heavy property of isotope is used by Meselson and Stahl to study DNA replication. Second thing these scientists used for their experiment is E. coli bacterium. These bacteria reproduce by binary fission, in which before cell division DNA is duplicated and then one cell divides into two. For E. coli bacterium, this total division process requires 20 minutes. So, after every 20 minutes, these cells will get doubled. If one bacterium is present at start, after one cell division cycle, it will produce 2 bacterial cells in generation 1, and after second cell division it will produce 4 bacterial cells in generation 2. Meselson and Stahl used this reproduction process of bacteria and isotope of nitrogen in combination, where firstly they have grown bacterium with N15, that is heavy isotope. So produced all bacteria will have heavy DNA, shown here in a orange color. Then this heavy DNA containing bacteria they have grown in another medium containing N14, that is light isotope of nitrogen. After one cell division cycle, 2 bacteria will be produced and their double-stranded DNA will have one old strand, that is N15 isotope containing, and one new strand with N14 isotope. These hybrid DNA containing bacteria, they have grown further for one more cell division cycle. Now, new bacteria will have N14 strand and 2 bacterium will have hybrid strand, and total 4 bacterium will be produced. To confirm this generation of light, heavy and hybrid DNA strands in each generation, Meselson and Stahl did density gradient centrifugation at each step. In first step, they have got DNA band near to the bottom of tube. It contains N15 isotope, that is heavy isotope. In second step, that is in first generation, they got DNA band in the middle of tube as it contains hybrid DNA with both isotope N14 as well as N15. In third step, that is in second generation, bacteria found are of two types. One band which is hybrid as that of second step and one containing light isotope at the top of the tube containing light isotope, that is N14. So these results proved that DNA replication is semi-conservative.